Bienvenidos. Meu portuñol no es bueno, pero tengo que cambiar para inglés. Professor Esplugas has made an excellent talk, and many of the aspects that I will talk overlap a lot with mine. He already has shown some of the international positions regarding biosimilars. So I'll make a quick sum up, and end up with a big picture of what I guess would be opinion of the global international community. So, in fact, biosimilars are exploding, and namely on monoclonal antibodies. But one of the major aspects that has driven some doubts on biosimilars came from erythropoietin, from the originator in the end of the 90s. The episodes of some pure red cell aplasia that occurred when there was a change in the manufacturing process was with the originator, in fact, not with a biosimilar. And these are the main doubts that are in the international community. Are they safe and efficacious? Can we extrapolate indications, as Professor Esplugas told? Can we interchange biosimilars with the reference drugs? And another question that is going beyond this, because we are going to have many biosimilars for the same infliximab, for the same etanercept. Can we interchange between different biosimilars? It's another question also. And what are the long-term consequences of these immunogenicity disparities? And are the requirements of the regulatory authorities enough to answer these questions? Okay, so I guess it's a matter of confidence also, not only for rheumatologists, but for hematologists, for gastroenterologists. The confidence we have on the studies and how they are produced, it's very important. So we all know the definition of biosimilars, and the first guidelines from EMA are from 2005, more than 10 years ago. It was the first international regulatory agency to regulate the biosimilar internationally. But the guidelines for the production of biosimilars of monoclonal antibodies was only released in 2012. Also, the World Health Organization and the FDA published some guidelines, and the common principles are about the same. To prove that the variability that is inherent to the production of biologicals stays within acceptable intervals and does not affect efficacy and safety when compared to the originators. And what do agencies demand that the pharma companies do? They have to do functional assays to prove that chemically they are similar, binding to antigen, Fc gamma receptors, complement, and minor differences are acceptable, as you know. But also, to prove biosimilarity, there has to be clinical studies, as you know. And to provide traceability, because they are just similar, they are not the same, it's really, really important that we are prescribing a biologic. We prescribe by either trademark or a code in the suffix in the end, so that we can, in the vigilance, the normal vigilance of clinical practice, have adequate traceability and have a thorough pharmacovigilance. And the first position papers, of course, on biosimilars were very, very generic, with concepts that we are not yet, that probably didn't exist. The concept of automatic substitution, interchangeability, was not even pointed out by these papers that have more than 10 years ago by the nephrologists when the first biosimilars appeared on erythropoietin. And we made a review of the the international community positions in 2014, now 4 years ago, and these were the position papers that were reviewed. 9 medical colleges or societies, 4 non-governmental organizations, 8 pharmaceutical organizations, 4 governmental departments, 4 pharmaceutical manufacturers. So a very broad range of institutions, of entities that have interest in biosimilars. And we went to see what happened. At that time, we in fact have had only 2 published trials at the time. The Planetas and the Planetra trials of infliximab, first biosimilar that was approved, the CTP13. So, what was the major position at that time? Regarding automatic substitution, it was mostly regarded as unacceptable. And if so, because the automatic substitution, which means that it can be substituted without the decision of the prescriber, as you have heard before. Well, if the regulators, if the governments want to impose that, because that's what it means, well, at least we have to do it by brand name in order to maintain traceability. In fact, these positions are not mutually exclusive, okay? Many of the positions, if they considered unacceptable, but if it is an imposition, a political imposition, and probably that's what's going to happen, well, at least we do it by brand name. But interchangeability, and interchangeability, it's a concept that's mostly developed by the FDA. It means that if you prescribe infliximab, it's the same whether you change it multiple times from infliximab originator to a biosimilar, then back to the originator, then back to another biosimilar, okay? The same efficacy, same safety, okay? Even though if it is imposed, most of those entities that I've shown you before, at least it has to have a consent by the physician. So, okay, imposed, but with some rules, with the consent of the prescriber. But some of the entities also suggested that there would be a patient-informed consent. So the patient would know and participate also in the decision. And the extrapolation. Well, at this time, we only had the studies of infliximab in rheumatoid arthritis and ankylosing spondylitis. As you know, it's approved by many other indications. And there was some caution with that at that time. We are talking about 2014. So automatic extrapolation, only 10%, a case by case, 30%, and against the extrapolation, 17%. Okay? The others had no position at all. And regarding immunogenicity and adverse events, so 60% would defend that robust pharmacovigilance must be assured, and almost 30% that immunogenicity should be adequately assessed. So, well, what was our position at that time? That drug selection should not be only based in economic aspects. Okay, there are other aspects that may influence our decision. As you may well know, for instance, in spondyloarthritis, we are choosing a drug not only by the first line. For instance, when we have extra-articular manifestations in a spondyloarthritis patient, we wouldn't be treating him, for instance, intestinal manifestations, we wouldn't be treating him with etanercept, even though we had a biosimilar infliximab that would be cheaper. So, besides, automatic substitution was not considered unacceptable after reviewing the literature at this time. And if the substitution would be politically imposed, we suggested that at least stay of a minimum of 6 months in the same drug in order to guarantee some stability under the same agent. Okay? And at that time, we had few data on extrapolation, and our position at that time was that extrapolation, so if a biosimilar was not tested for inflammatory bowel disease, for instance, we wouldn't use it if it wasn't tested. That was our position at that time. Regarding the evidence available, I reminded you, at that time we had only 2 published trials on infliximab biosimilar. So what was the position of other societies? For instance, just as an example, the Italians had made a joint position at 2014 regarding rheumatology, dermatology, and gastroenterology. And well, they were a little more evasive regarding extrapolation. Well, biosimilars still need to be tested for efficacy and safety by well-designed trials with adequate sample size, especially for psoriasis and IBD, because we had only trials for rheumatoid and spondyloarthritis. And caution is recommended for extrapolation. And the Spanish position paper was also a bit cautious, but, well, extrapolation would be acceptable according to the EMA standards. Was a bit more evasive than the previous positions. Well, we reviewed our position regarding the evidence, regarding the appearance of, for instance, Northwich, that was already thoroughly explained. And now, in our recent position paper in 2017, extrapolation is now acceptable, but interchangeability not, and automatic substitution also not acceptable, but judicious switch is desirable. Well, we are also taking into account that probably automatic substitution may be somewhat imposed by the regulators, which already in some way has happened in our case. Our Infarmed, which is our pharma national institution, has already issued a norm suggesting that we can change all the approved biosimilars in all patients, with some regard in an individualized decision, but trying to push that decision forward. The dermatologists are more conservative. This was from 2016, and they are clearly against switching from a reference agent to a biosimilar. This should not be recommended, and any decision regarding automatic substitution is strongly objected as the major position. But they are a little bit more conservative than rheumatologists, and they also state that biosimilars should not prevent or delay access to therapeutic innovation. And what is the position of our pharmacists, which is curious. And broadly, they put this interchangeable concept into play, which is not clearly defined by EMA. EMA puts the decision on automatic substitution to each member state, as already Professor Explugues explained. And, well, they say that if a biosimilar has proven biosimilar, it's inherently interchangeable, which is a very, I wouldn't say, which is not unanimous, as you may agree. So, due to minimal time of anti-drug antibodies, development of good clinical practice, the interchangeability among biosimilar and originator is not recommended during the first 9 months of therapy. Well, we have defended that at least the patient had to be at least 6 months. So they say that biosimilar and interchangeable are almost synonyms, but they protect that position by saying, well, at least the patient has to be stable 9 months before we do a switch, automatic substitution from an originator to a biosimilar. So, well, I won't lose time with these slides because the differences of the different health agencies were already exposed by Professor Explugues. But mostly interchangeability, it's a concept. And there are norms published by the FDA on that. And usually to prove interchangeability, in fact, it's more difficult than just to prove biosimilarity. Okay? And the trials that have to be done to prove interchangeability have to have this design. We are stable on a reference on the originator, and one group stays on the originator, the other makes one first switch, then another switch back to the originator, then another switch back to the biosimilar. And that's the demand of multiple switching to say that this product is not only a biosimilar, it's also interchangeable. Okay, so we have to have multiple switch. We already have some trials with this design published. We have the Egality trial on psoriasis, which does multiple switches between etanercept originator and a biosimilar of etanercept, and with the same result, in fact. So in psoriasis, we already have this evidence of an interchangeable, if we want to say, product. There are some countries that already have given green light to switch some stable patients. We already have done that with the infliximab, as Professor Eurico Fonseca will show in the next presentation. Although they do this green light, the position stays already in the treating physician. And regarding automatic switching, still automatic substitution, still the dominant position is no yet, or if yes, at least with the consent of the physician, which in fact it's not an automatic substitution at all. It's not the concept of automatic substitution is the decision is outside of the physician, in fact. So this is my last slide, and then what I think that are the dominant positions internationally. Well, in fact, economic reasons shall not be the only reason to take into account when we prescribe biologics. We have other aspects of disease that we can use, and we can use that to convince our institutions to prescribe in a certain patient this or that biologic. Okay. And extrapolation may be acceptable in most indications, but with some reservation in some indications and some specific patients. And interchangeability may be acceptable under physician supervision and with consent after a period of stability, to avoid that patients do multiple switches with different biosimilars. And we have to think of interchangeability a little away from that. We have that design that we compare a biosimilar with the originator. But we are talking about interchangeability probably between different biosimilars. That's not going to be done, probably, because it would be economically not reasonable, and would remove the potential benefit that we have to have by having more economic biosimilars. So ideally, biosimilars should prove interchangeability, but at the extent of the reasonable. And that would lead to the consideration of considering, well, if it is interchangeable, we would accept better the automatic substitution. Because if it is interchangeable, well, You can do multiple switch without losing safety and efficacy. Okay, and at last, we have to have good registries, and we have to have the capacity to differentiate when there is a change from the biosimilar to the originator and back to the biosimilar and vice versa, so that we could have a good traceability in order to avoid mistakes in pharmacovigilance. Okay, thank you for your attention.